Hello students. So next point in your syllabus, that is the theta models. The question asked in exam, explain the theta model, or most of time these are called the short notes. So previously I talked about the prokaryotic cells. So in prokaryotic cell there is a circular shaped DNA. So in that, in E. coli there is the parental DNA remains in circular form throughout the replication cycle. So your prokaryotic cells or in E. coli, in, in replication process or in replication cycles, the DNA will become the circular shape throughout this cycle. So during replication, in this DNA there is the bubble formation is take place, and due to these bubbles, the this circular DNA makes the theta shape. So these two growings are established, and replication is the bidirection. So at the two sides the replications will be start. Then these both strands is replicated, but as the two DNA strands in a circular circulars are uncoiled for the replication, it creates the uh, coiling tensions. So the DNA molecules become the supercoiled, but the enzyme topoisomerase 4 are helped to remove the trace from this DNA supercoiling. So these are happening in case of the prokaryotic cells or in E. coli. Due to the in that cells there is the circular DNA is present, and in the replication process the replication bubble is formed, and these bubbles create some, or these bubbles are initiated these replications. So these DNA, circular DNA creates the shape like theta models. That's why this stage or situation known as the theta model. रेप्लिकेशन जो प्रोकैरियोटिक्स में आई ऑलरेडी टॉट अबाउट द रेप्लिकेशन इन प्रोकैरियोट. सो इन दैट देयर इज द सर्कुलर डीएनए रहता है, फिर वहाँ जो ओराइसी रीजन है वहाँ पे रेप्लिकेशन स्टार्ट होता है. व्हेन दिस रेप्लिकेशन इज स्टार्ट फर्स्ट at that stage there is the formation of replication bubbles. So in that replication bubble some traces are also formed on the other DNA strands, and these replications are starting at the both end, or it will be bidirectionally it happens. But overall the replication process, these DNA are in circular form, and they are formed the two new DNA strands from one old strand. So this process is happening in prokaryote and also in E. coli. So these theta models are observed in all the prokaryotes and E. coli and those cells who contains the circular DNA. So I hope and sorry, you have to mention this figure. So in that figure we shortly mention that this replication process, and in that This DNA is look like the theta structure. So in exam, most of time these ask for the short questions or short notes. So I hope you got this theta model. Now I stop here.